Hello everyone, my name is Dana Al-Hamdan, a student in Al-Ain University. I worked with my colleagues Hala Al-Khatib and Marina Anasrallah under the supervision of our professor, Dr. Mohammad AlAhmad. We are going to discuss the clinical application of pharmacogenomics in the treatment of bacterial infections. In our research, we presented a personalized prognostic tool that will further improve the management of bacterial infections. We are going to discuss the bacterial genome, the host genome, and finally present our personalized prognostic tool. Pharmacogenomics is a field that was successfully applied to some diseases like cardiovascular diseases, and the basis for such applications is performing genotyping for patients to customize treatment regimens according to their genes. The application of pharmacogenomics in bacterial infections is slightly more complex because we need to consider the bacterial genome in addition to the host genome. Therefore, we need to identify the polymorphisms in the drug metabolizing enzymes and drug transporters that alter the pharmacokinetics of the antibiotics we intend to use. In addition to that, we need to identify the polymorphisms that affect the immune system, which explains the inter-individual variability in the response to the same infections. On the other hand, the bacterial genome needs to be taken into consideration by identifying the resistance genes that help bacteria bypass the effect of antibiotics. All these genetic factors are important determinants in the prognosis of the disease and the treatment outcome. What we did was we summarized some of the genes that are responsible for antibiotic resistance along with the mechanisms of resistance. With the help of bioinformatics, bacterial genome sequencing could be performed in order to aid the process of antibiotic selection. In other words, we can avoid the use of conventional antibiotics to treat such infections, since these resistance genes will render them ineffective.
Now let's move on to talk about the host genome. We'll talk about the pharmacogenomics and immunophenotype. Antibiotic agents have dramatically altered the history of infectious disease. They significantly contribute to improve healthcare and clinical practice in bacterial infectious due to the high reduction in rates of mortality and morbidity. The implication of pharmacogenomics in clinical practice of infectious disease area is associated with a complex relationship between the drug's pharmacokinetics and the immune response of the host and bacteria. So, regarding the pharmacogenomics, we will talk about the pharmacokinetic variability factors. We have two factors, which are antibiotic-drug interactions and ethnicity. For the first factors, antibiotic-drug interactions, co-administration of antibiotics with other therapeutic agents may result in alteration in some enzymatic activities, such as CYP450 enzymes or a transportation system activities such as a V-glycoprotein in the host. And regarding the ethnicity, distinct ethnicity might play a role in the genetic difference and therefore in the clinical response to the therapeutic agents. For instance, varied ethnic groups might have differences in drug transporter systems, such as a P-glycoprotein or CYP450 drug metabolizing enzymes. And now, moving on, we will talk about the single nucleotide polymorphisms. Importance of identification of the single nucleotide polymorphisms. The expression of host enzymatic system or a transportation protein of an antibiotic might be exposed to varied alteration, which influence antibiotic pharmacokinetics. This can be identified as a single nucleotide polymorphism. In several genes coding drug transporters, like for example, uptake or efflux transporters and enzymes, single nucleotide polymorphisms have a vital role in such process. Now, we will talk about the CYP3A4. One of these enzymes that is significant in the area of bacterial infections is the CYP3A4 enzyme, which was found to be involved in the metabolism of numerous antibiotics. Regarding the selection of therapeutic agents, identifying the polymorphic forms of this enzyme will provide a better approach to antibiotic selection. The selected drug must be adjusted according to the respective enzyme activity.
I'm going to talk about immunophenotype. The inter-individual differences in the immune-mediated response to bacterial infections and susceptibility to pathogen infections are substantive and can ultimately lead to treatment failure. Studying genetic determinants have contributed in launching personalized medicine genetic approach. The innate immune response can be affected by how bacterial ability to manipulate the expression of particular gene. For instance, some infectious bacteria are identified in nucleomedarins due to their ability to manipulate the transcription and epigenetic process by evading the nucleus of the cells. Understanding the immunophenotype of both the host and pathogen is the key to content and manipulate with their genomes and determine the antibiotic treatment outcomes. The interaction between the host and the bacteria during the infections is complex. Immunophenotype variations are dramatically increasing this complexity. Application of immunopharmacogenomics. Translation of genomic sequence of a pathogen into an individualized therapeutic approach can be applied through identification of immunologically important peptides for what is known as cytotoxic T lymphocytes epitopes, which are considered critical for a specific immune response of a host. Although the application of immunopharmacogenomics in clinical practice is still pending due to its complexity, it's a promising approach that will improve the management of infections. Immune response variability factors, the human leukocyte antigen, and the human evolutionary biology. Given the frequency of genetic polymorphisms between populations, we can expect marked difference in immune response between individuals. Therefore, it's important to incorporate immunopharmacogenomics into the treatment of infectious diseases. Genes of a human leukocytes antigen system, T cells and B cells, are all key factors in determining immunophenotype. Complex mutation in the human leukocyte antigen system result in high variability in T and B cells. Polymorphism of HLA class II determine the response of bacterial antigens by influencing the cytokine response. Nowadays, it has become easier to perform DNA sequencing to identify the different groups of T and B cells. Although the application of immunopharmacogenomics in clinical practice is still pending, it's a promising approach that will improve the treatment of infections. Several levels of environmental factors coupled with genetic determinants can influence immunophenotype variations. This considerably augments the immune response variations between the host and produce variability in adverse drug reactions against different antibiotic treatments. Moreover, the long biological coevolution of the host cells and the pathogenic bacteria has been developed with different levels and cause substantive biological alteration due to evolutionary factors such as genetic, drift, and migrations. Some genetic alterations have positive effect at the immune system and the defense mechanism, which can implicate protection. We will propose a personalized prognostic tool that predicts the severity of infection and help us in choosing the best antibiotic.
When it comes to the host genome, the polymorphisms in genes responsible for drug metabolizing enzymes and drug transporters need to be identified. Like for example, as we can see here, for the drug metabolizing enzymes, we have the CYP3A4 polymorphisms. And for the drug transporters, we have the ABCB1, ABCC2, and solute carrier polymorphisms.
In addition to that, polymorphisms in the immune system, specifically in the human leukocyte antigen system, need to be identified. So to summarize everything in this table, we can see that the polymorphisms in the drug metabolizing enzymes, drug transporters, and HLA class 2, it will affect some antibiotics, such as erythromycin, tetracycline, levofloxacin, penicillins, and linezolid.
Finally, we came up with this algorithm to treat bacterial infections, and this begins with performing genotyping for the patient to identify the polymorphisms that affect the pharmacokinetics of antibiotics and the variations in immune response discussed previously. Next, bacterial genome sequencing with the help of bioinformatics must be performed to identify the resistance genes that a bacteria may carry. Based on that, we divided the treatment options into 3 categories. If the host carries polymorphic genes and the bacteria carries resistance genes towards a selected antibiotic, then this treatment option should be avoided, as it will be ineffective. Next, if the host carries polymorphic genes, but the bacteria is not resistant toward the selected antibiotic, then this is a possible treatment regimen. Finally, if the host does not carry a polymorphic gene and the bacteria is not resistant towards a selected antibiotic, then this treatment regimen will be ideal, and it is recommended for the successful eradication of the bacteria and better management of the disease.